All right, great. I see most of you guys have finished that. Thank you so much for answering that. I'll leave it up for just a little bit longer for those of you who are still working on it. I wanted to introduce our second speaker. Next we have up, I'm going to botch her last name. I'm terrible at this, so I'll let her pronounce it too. Maria Hadjifrangiskou, who is an associate professor of pathology, microbiology, and immunology at Vanderbilt. And she's the associate director of the Vanderbilt Institute for Infection, Immunology, and Inflammation, or VI4. And Maria is just a delight. And so she's going to tell you also about her background and personal story. And let me pull her up here. She said that her kid might come up. We might be able to see her children. We're all dealing with being at home through this. So that would be wonderful. So here we go. Maria, I see you're here.
Yes, can you hear me now, guys?
Yeah.
And can you see me?
Okay. I see some thumbs up, but I can't see. Oh, there you are. Hi.
Yep. Hi, everybody. All right, so if you let me share my screen, I can put up my presentation. I'm super excited to be with you all today. I love having students through VSSA, and I'm excited to tell you my story. And I'm sad to see that Ben left the building because my cat is 18 years old, and and he's faring in good health. All right. Can I share my screen now?
Be good to
Yep.
go. Thank you so much.
Sure. Let me see if I can do that real quick. There it is. Bear with me, guys. Yep. Let's see if you guys can see that. Oops, sorry. Can you see these guys? Yeah?
Perfect. Thank you.
Awesome. All right. So just like Ben previously, I'm here to tell you a little bit about my journey to my scientific career. Like Beth said, I'm associate professor here at Vanderbilt at the Department of Pathology, Micro and Immunology, which is a blended department that focuses on those three areas of research. And I'm a bacterial geneticist, so I fit in the realm of microbiology. But I didn't know that that's what I wanted to do right when I was your age and earlier. So I'm going to give you a little bit about my journey, and then we'll return to what you're looking at here. So I'm not from here. I was born on this little beautiful island in Eastern Mediterranean. It's called Cyprus. It's an island nation. These, what you see here are my toes in one of our wonderful beaches. And this island is rich in very, very long history because it sits right at the interface of three continents, Asia, Africa, and Europe. And even though the island is magnificent in terms of having a wonderful summer vacation, at the time I was growing up, there were not many opportunities for an education in science. So for me to study science, I had to get off the island on a scholarship. So why on a scholarship? Let's back up a little more. I was born to a very happy but not wealthy family. The island of Cyprus was in a war in 1974, which divided the island and led to the formation of thousands of refugees. My dad at the time, who was a high school dropout and in the police academy, was the only one of 7 siblings that was not displaced because of where his house was. So my mom and dad at that time actually had to house 70 family members of my dad's family for a year and a half, which devastated their finances and put us into below blue collar family. My dad was at the police academy. My mom actually was a nurse, and she was a nurse for 33 years. So I was born 3 years after the war. My displaced family was now in some sort of refugee housing. So I only knew my grandma and my grandpa living with us. And like I said, we were like super happy, but we didn't have a lot to live on. So, like, for example, on Thursday nights, our dinner would be grilled cheese toast. And it was my favorite dinner because we would eat it and watch Premier League soccer that was streaming from one channel that we had on a TV. So for me, it was a very fond memory, but little did I know at the time that we were having grilled cheese because that was the only thing that they could actually afford. And also, with the fact that we only had one TV channel, I was mesmerized right off the bat with documentaries that are super old for now. This was like stopped airing in 1976, and in Cyprus it was showing when I was like 5 years old, right? This was the underworld of Jacques Cousteau, who was actually a marine biologist, and I was just fascinated watching this person really investigate what was under the in the ocean. And that, I think, was my very first interaction with the science, and it was the first time I realized that, oh, this is super cool. I was always very curious. I always wanted to know how things worked. I'm showing these pictures here. On this side, you see an olive tree, is native to the island of Cyprus. I actually set fire on one just to see what happens. I put here this red race car because my brother, I have 3 older siblings. They're much older than me, 8 years, 10 and 13 years. This was the youngest of the 3 siblings' car. I actually pulled it apart to know how it works and whether I can put it back together. When I started school, I could not sit still in didactic lessons. I was bored with everything else except with the science classes. And when I entered high school, that was the first time that I actually really truly fell in love with the concept. And that was when a professor suggested this book, Microbe Hunters. So I devoured the book. I fell in love with the concepts of having these early day microbiologists and how they studied microbes and how they developed vaccines. I knew then that I wanted to do something in that realm of biology, but I still didn't know what I really wanted to do and what it really meant. Also, like I said before, my family was not affluent. So in order for me to get off the island, I had to find a way to finance my future. So I applied to a bunch of scholarships. One of them was a Fulbright scholarship. I did that at the beginning of my high school senior year. I wasn't sure or hopeful that it would hit, but it did. So I was awarded one of the 14 Fulbright scholarships that were on the island. And then I was tasked with a decision of finding where to go. So my dad and I literally took this college catalog from the United States and looked around to see, A, what universities were there. And there were so many, we were overwhelmed. And then we decided, you know what? Let's just decide based on the 4 seasons. So, totally serendipitous, right? We had zero guidance. Like my dad was a high school dropout, right? So we chose Clarion University of Pennsylvania because it was in Midwestern Pennsylvania and it looked like it had 4 seasons, believe it or not. So in 1996, with 2 suitcases and my dad, I landed in Pittsburgh. He took a loan to come with me and come to Pittsburgh. And I don't know how many of you are familiar with Western Pennsylvania, but there's a specific slang in Pittsburgh is, I learned about chipped ham. I learned my first interaction with really mainstream English in Pittsburgh. And of course, I'm a hardcore Steelers fan because of that. So Clarion University is located 90 minutes north of Pittsburgh. It's a beautiful campus. Like I said, I wanted to see the 4 seasons, but this is what Clarion looks like from October to May. And through here, I didn't only learn that, I'm in a new country, in a huge culture shock. I also learned that not many people knew where Cyprus was. So I was asked questions like, do you wear jeans? Are there butterflies in Cyprus? Do you guys live in huts? All these are questions that I was asked, and I was incredulous. But at the same time, I realized how little I knew about the culture I was thrown in. For example, I thought, up until I arrived in Pennsylvania, that Amish were not real. I thought that they were a figment of Hollywood until I saw them in Western Pennsylvania, and I thought that somebody was making a movie. And then my friends there actually let me know of the facts. So, like that began my journey in essentially learning not things about science, but also blending myself into a new culture. And it was very difficult at first because my English were not as good as they are today, but I learned from those experiences and I value them to this day. And I got my first real experience in microbiology and in mentoring from this person here who still teaches at Clarion University. His name is Dr. Doug Smith. He exuded enthusiasm. None of his classes was boring. I was actually tasked to be part of a movie explaining host pathogen interactions. We had to make a James Bond movie to explain to him our understanding of molecular microbiology and microbial pathogenesis. And through that experience, through that hands-on interaction with my peers, and through witnessing how excited this man was to come in the class and teach us all he knew, I realized, A, that I really wanted to affect people the same way he did, and I also started charting my path. I really started distilling that, oh, I really want to know how microbial pathogens influence health and disease. So after I graduated from Clarion University in 2000, I took a year off because I thought I would go back home and chart my path there. But after having these experiences that I just described, I took a year off. I worked as a lab tech at Quest Diagnostics for a year, took my GRE, and applied to different graduate schools. And one of the schools that really I focused on was UT Health McGovern Medical School. And the reason I chose to go there was this woman here on the right. This is Terry Kohler. She was one of the few people in the United States who actually worked on anthrax. So after reading Microbe Hunters early on, and after going through the microbial pathogenesis coursework that I talked to you guys about, I realized that I really wanted to go into infectious disease. And naive as I was like, what's more infectious than something like Bacillus anthracis that causes anthrax? At the time, this was before the 9/11. So actually, I joined her lab about a month before 9/11. And what you see here is a Getty image of her, because once the anthrax attacks occurred, our lab became famous like every other lab that was working on anthracis. And here is my little Getty image. I'm here at the background. I'm actually rotating in the lab when they're taking these images, and I'm taking something out of the freezer there. But most importantly, through these experiences, I spent 6 years in Houston studying bacterial genetics and how Bacillus anthracis controls the production of toxin and capsule, which are the major virulence factors that cause anthrax disease. And I really came to the stark realization that even though this pathogen is super interesting, the burden of working in a biosafety level 3 environment was too much. So I wanted to change direction for my postdoctoral work, and I decided to go away from select agent research and try something different. Through the leadership of Terry as well, I realized that I wanted to do what she does. I wanted to be able to mentor students. I wanted to be able to write the grants. I was thrilled about making discoveries. So it wasn't until the 3rd year of my grad school career that I made these realizations. So don't worry, guys, if you're at this stage in your life and you don't really know what to do, just follow your heart. as you've seen so far, like every decision I made was partly serendipity, partly I was following what I really wanted to do, what made me excited. And I don't regret that one single bit. Also, I started low, right? I didn't go to MIT for my bachelor's degree. I went to Clarion University of Pennsylvania. You can work yourself up. Don't always have confidence and follow your dreams. I can't stress that enough. So from Houston, after I graduated with my PhD degree, I sought a postdoctoral fellowship at Washington University in St. Louis, where I was there for 5 years. I did my work on pathogenic E. coli, the E. coli that causes urinary tract infections, and we're going to go a little bit over what my research team works on now. But what I wanted to stress here is that the mentorship of this man here, his name is Scott Hultgren. He's a National Academy of Science member. He's a leader in his field. When I joined his lab, there were 24 members in the lab. 14 of us were postdocs. And I don't know how he was able to keep up with all of us, but one of the biggest achievements unlocked in that postdoctoral career was actually the ability for me to develop my independent research plan with his support, learn how to competitively write, but most importantly, develop a support network in my scientific siblings. It is very important to form bonds and have a network that will support you and be there for you and be there with you through the ups and the downs. These are only a few of them. We see each other in meetings to this day. We fall back on each other when we have hard times. I cannot stress enough how important it is to have sense of family. And one of the biggest reasons why we have that coming from that lab is that Scott leads from the bottom up. We were all part of decisions. Nobody was left behind, and leadership was a team effort. He is humble as a leader, and it is one of the most important attributes that I value to this day. So networking is key. I got 4 invitations for job seminars from a talk I gave at a Gordon Research Conference. And from multiple job offers, I accepted Vanderbilt, again, because I could see that support, both for faculty and for students, was a priority. So I set up shop here. We study uropathogenic E. coli, which is the agent of urinary tract infections. This is my team here through Zoom. And I want to point out that this here that is highlighted is actually an alumnus. And over here, this guy right here is an alumnus, and the one person behind them. So we still have people who join in who have left. And it's an amazing network, because that's what I really valued from my training, and I want to pay it forward. So the current team is these guys over here: Seth, Connor, Grace, Ham, Melanie, John, Thomas, and Michelle. And we all together collectively are excited about understanding how bacteria build their cities. So these are called biofilms. It takes one single bacterium to adhere to a surface like a catheter, and then it expands both on the 2-dimensional and the 3-dimensional surface. And as you see here, we have different colored pieces of this multicellular puzzle, and this is to show that we have different genetic programs producing different material as the bacteria form these multicellular cities. And if you equate this cartoon on the left that you see with an actual bacterial colony, what you see on the right is a uropathogenic E. coli colony, and on the top here is a colored drop of water. And what I want to instill in you is that when the bacteria make these cities, they essentially cocoon themselves in material that prevent diffusion of water-based solutions such as antibiotics. So if you have bugs that are forming these multicellular cities, you cannot attack them with antibiotics, even if on the single cell level they are susceptible. So my team really wants to understand how these cities form and what we can do to dismantle them. Let me see if I can move this forward. There you go. Oops, sorry guys. So why do we want to do this? Because in uropathogenic E. coli, the formation of these multicellular cities is critical for infection. Here is what I hope you can see my arrow, guys. What you see here is a single cell, and these hair-like naps are what called are adhesive fibers. The bacteria express 4 to 14 different types of these, and these little tips at the top actually allow the bacteria to recognize different receptors on the host cell and allow the bacteria to bind. When the bacteria bind, they get internalized and they form these biofilm communities inside bladder cells. So here's the cartoon, and on top here, this bacterial community that you see labeled in green is actually inside a bladder cell. And then as this bacterial community expands, it can sense that it doesn't have food and water and oxygen, and it actually morphologically changes, and now the bacteria can filament, extrude out of the cell, killing it, and this vicious cycle can go over and over and over. In the genitourinary tract, you also can have bacteria colonizing vaginal epithelial cells, and in this case we can have bacteria being both intracellular and extracellular, and these are images that we take in the lab trying to understand the architecture of these bacterial communities so that we can better attack them therapeutically. And what you see here in micro image, we can actually identify in the urine of patients with acute and recurrent urinary tract infection. And in our catheterized patients in the hospital, you can have biofilms forming on the catheters, and we can zoom in on these biofilms, and we can again see that the bacteria are cocooned in this material they produce that then prevent antibiotic treatments and the immune responses from getting to them. So this is one of my students, Connor, and it's displaying this in real time. So this is again a colored antibiotic droplet, and you can see there's no seepage through. So we're working to find genes that are important for making this cocoon so that we can actually better attack these communities. On the right hand side here, what you see is a confocal image of bacteria that are in the biofilm, and I hope you can appreciate that the treated with antibiotic community looks no different than the untreated, unfortunately, and the number of bacteria that remain alive does not change. So just like Ben, I'm going to refer to an old movie called The Matrix. I'm a huge sci-fi fan, but just like Neo in The Matrix, the biofilm matrix that these bacteria make blocks any external stressors. And we know a lot about what it's made out of. It's two different components that are the primary agents of keeping soluble agents away from the bacteria. These are called Chiari amyloid fibers and cellulose. And we're trying to understand how are these common goods distributed over time as the biofilm communities form. How do the bacteria coordinate the expression of these common goods? And like I said, how can we break the system to prevent formation of these pathogenic communities on catheters and within human hosts? And with that, I'm going to stop there. I have all my info. Beth has it. You guys can follow us on Twitter at Bacterial Talk. These E. coli strains are called extra pathogenic E. coli, or XPEC. So we have a hashtag Great Expectations for when we tweet out news about our lab. And feel free to email me. I can answer any questions you have and chat with you at any moment you want. And again, I'm very happy to have been a part of this. And I'll stop share right here.
Thank you so much, Maria. What a wonderful, just incredible journey you've had. And I so appreciate that all of your dedication and perseverance to be where you're at. Thank you so much. we've got lots of questions. Do you have a few moments for that?
Sure. Yeah. Like I said, a kid might pop up, but I'm very happy to answer questions, of course.
Awesome. Okay, this is a really good one. What strategies do you use when you come up with your research plans and aims and action plans?
Oh, that's a great question. So actually, I constantly exercise the concepts of rigor and reproducibility. So every time we have a new crop of students, we do a couple of things that help me reset as well. One of them is something that actually Beth already knows about. There is this awesome online resource called iBiology that takes students through a process, very simple process of let's get started experimenting. And believe it or not, every time I have a research question, I go in and do those exercises. And essentially it allows me to go like, okay, I'm excited about this. And I'm one of those mentors where I really get excited, man. And I just throw these kids like questions. And then I'm like, okay, let's prioritize. So through this tool, I put down what the big question is. And then I try to really break it down into, okay, what's the big picture? What are the sub aims are going to be, and are they helping me get to the big picture? And then what are 3 or 4 solid experiments that we need to do to address it? And then I kick it to the team, and the team decides with me, hey, this is a good experiment or this is a bad experiment. And then we think about why, and then we put down the controls and we go on. It's a long process, right? And there's a lot of trial and error, but that's what's fun about research, at least for me. Does that answer the question?
That's awesome. And thank you for mentioning that iBiology series. I'll post a link to that for anybody
Yeah.
who's interested. It's a great way to kind of stepwise your way through project development.
Mm-hmm.
Okay, another interesting question. What was it about a BSL-3 lab that made you not want to continue in there?
So the BSL-3 itself is not, it's, I'm very appreciative that I learned how to work in a BSL-3 level containment. So essentially you have to gown up, there's an antechamber, you go in, you gown up again. And for example, right, I could do an RNA extraction experiment in an hour and a half outside in the BSL-2 lab. It takes you about 4 hours in the BSL-3 lab. Because when you work with something like anthracis, which is a spore former, and the concern is that you could bring outside something that was biologically active, you have to make sure that everything is inactivated before you come out of the biosafety level 3. And even though it was super exciting, right, guys, if you notice if you go to my website and you pull down my CV, or if you PubMed me, you will see that I have 2 papers from my grad school career, and you will see a 3rd anthrax paper that came last year, because all my work was in a fully virulent strain. And when the anthrax attacks occurred, we couldn't publish that anymore. So we had to wait. And you can see that my work took 9 years. I was very proud of it. But that was one of those things that as a student, right, that was one of the biggest challenges, because I realized that I needed to be super competitive. And my even though my PhD training was fantastic, I didn't want to have those hurdles as a PI. I wanted to be able to train as many students as I wanted, and I wanted to be able to give them first author papers within a timely fashion, if that makes sense.
That makes a lot of sense. Another good one. How did you manage the stress of being in a different culture during a mentally demanding time like undergrad? That's a big transition at an important time. What strategies did you use?
Yeah, so honestly, at the time, I didn't know I was using them, right? So in the first 3 months, I literally smiled and waved, because I thought I knew English when I came, and I literally understood about 35% of what people were telling me. So that was very, very hard for me at first. And I was also, for the first year, I was offended with everything. But then I realized through my friends, mostly, I made some really, really good friends early on, and those ended up being my sounding board. They were the ones who said, hey, this is what they meant, because I didn't know figures of speech, right? I didn't know what it means. Like, I don't know, like they're selling like hotcakes. Like I would say they're selling like pancakes, right? I still to this day say things that don't make sense to people because I don't remember these things very well. But my biggest sounding board were making those friends and realizing which people were open to becoming friends with me and being patient with me. Does that make sense? And that really, really helped. And it also helped for me to break those boundaries and be vulnerable and open up with what help I needed. And that took about a good year to do that.
That's true.